My name is Edward Lanphier. I'm the President and CEO of Sangamo BioSciences. I first apologize for my voice. I don't usually sound this sexy. It's viral induced, and for this meeting that's supposed to be amusing. So thank you, mercifully. Good afternoon. It's been a very interesting morning with a lot of discussion, a lot of focus, I think, in really a lot of blocking and tackling towards commercialization on manufacturing strategies, regulatory issues surrounding those decisions in manufacturing, process development. This afternoon, we're going to take a bit of a turn from that and in several ways. First, we're going to talk a little bit about an area called genome editing, and I'll introduce that topic in a moment. But I think it's, as you'll see, it may be an area that begins to look at second, third generation strategies from our current thoughts about gene therapy and modified cell therapies. We'll then have a talk about large-scale DNA manufacturing, certainly something quite important and consistent with this morning's discussions. We'll then turn to a discussion of business models, looking at points of view from both large pharma as well as biotech. And then lastly, we'll have a panel on commercialization, and we'll be able to hear from uniQure and Christian Myers in particular, with the first and only product in terms of a gene therapy product on the market here in Europe. But we'll also get a perspective from pharma as well on that. So it's a good, interesting, packed afternoon, as often is in the afternoon sessions. We are what's between you and cocktails. So we'll try to be efficient. If nothing else, my physician told me bourbon would be helpful for this. And so we'll find out in about an hour and a half. All right, so let me start this with an introduction in the area. Actually, there was a couple of things I was supposed to read. I think the main thing I'm supposed to ask you to do is turn in the evaluation forms. And I know they'd appreciate it if you'd do that. All right, so first topic of the afternoon is the area of genome editing, and in particular, looking at this from a potential second, third generation strategy as it relates to current gene therapy approaches. And so there are a couple of ways to think about this, or a couple of ways to potentially slice the pie. And so I'm going to try and do that in three buckets, and then we'll have a panel to follow this with two other colleagues in the genome editing space, and we'll try and answer any questions you have. And if we haven't stirred up any controversy or questions in the audience, we'll try and stir up some of our own on the panel. So I'm going to start with a discussion of ex vivo genome editing versus sort of the current standard of art in the area of cell modification of lentiviruses. And then I'm going to turn to genome editing and comparing and contrasting that with AAV gene therapy approaches, particularly in the rare and orphan disease space, and particularly in those areas of dividing cells. And I'll try and make that point as we go through it. Then lastly, come back to a higher level discussion of different genome editing platforms and how those compare and contrast. And maybe we can stir up a few angry looks and deep controversy. All right, so start with the ex vivo side of things. So I won't go through each bullet point. There are several slides that have this as a basic outline. The slides, as I think most presenters are available after the meeting. But the idea here is to look at what are the principal issues that we think about when developing a human therapeutic in genetically modified cells, whether those are in progenitors or in terminally differentiated cells, CD4, CD8s, and so on. So the critical issue, particularly in progenitor cells, is that you have the ability of whatever modification you make, that transgene or knockout, whatever it happens to be represented in the differentiated cells. Otherwise, modifying the stem cell is great, But if you're ultimately trying to have a therapeutic effect in the differentiated populations, that's going to get washed out. That's going to be eliminated. So the critical challenge, the critical need, quite frankly, is modification in the endogenous genome in order that that characteristic be carried through in the terminally differentiated population. So these are some of the kinds of things that at least I think the panel think about when thinking about comparing genome editing strategies, which allow one to build a DNA binding element, target a defined sequence of DNA, and then modify that sequence either by disrupting the endogenous gene, so permanently disrupting or deleting sequences from that site, or by using that double-stranded break, and because of the precision of that break, to use that for targeted insertion or homologous targeted introduction of a defined sequence into that site. This is in contrast to a lentivirus, which carries in a therapeutic transgene, which is driven by its own promoter, but that integration is essentially random. I mean, we can talk about random and the issues of random, but it's essentially random. And given, as I like to say about these random integrating vectors, given enough monkeys and enough typewriters, you can eventually write Shakespeare. Given enough integrating vectors and enough cell types, eventually you're going to have an insertion at a site that can drive an oncogenic event and insertional mutagenesis. So the principal issue in terms of differentiation, and again, I'm not going to read through all these, is really one of targeting and the specificity that can be achieved with genome editing approaches versus the potential of insertional mutagenesis of retroviral or lenti-based vectors. So that's a compare and contrast, ex vivo gene modification of progenitors versus a genome editing approach. Similarly, we can look at this strategy for in vivo opportunities or in vivo genome editing. And one of the things about genome editing is it allows one to be relatively agnostic about the gene one wants to target. And so within the context of 3 billion base pairs, in the context of 30+ thousand genes in the human genome, differing platforms, differing genome editing platforms have the ability to target sequences in a relatively agnostic way. And so if you can do that, one can think about going in, targeting mutations, and literally, physically, permanently changing DNA sequences in a way that can drive a mutation to a wild type sequence. And so if you think about things like sickle cell disease, where there's a mutation in the beta globin gene, one can think about modifying that nucleic acid mutation in a way that corrects the sequence. So that gives one the ability not only to go in and permanently modify an endogenous gene, which then gives you permanence within that cell type, but it also gives you the ability to control that expression potentially under an endogenous promoter versus an exogenous promoter. So as you think about current approaches with AAV, there are many, many strategies that I think collectively, I certainly think are going to be very successful. I think some of the challenges that we consider in most acute are in dividing cells, so not in the brain necessarily, not in the eye necessarily, but in the liver. And this slide is principally focused in areas of replicating cells. And so the major issue we'd highlight here, although I think there are others, is in the issue of durability of an episomally retained vector versus correction or insertion into the endogenous genome, particularly in the liver. So lastly, just to move to the current major focuses in the area of genome editing, there are 3 platforms that are currently under significant development and quite frankly, significant investment and continued intellectual and scientific evolution. Those are at the far left, the zinc finger nuclease platform. That's the work that I've been most involved with. There's also TALENs. TALENs and zinc fingers are protein-based approaches and are found or can be engineered in that way. And then the Lewis kid on the block in the area of greatest enthusiasm, I think, and visibility is the area of CRISPRs, and we're going to hear from 2 panelists in that space and give us some sense of that. all of these approaches have their strengths and their weaknesses, their pluses and their minuses, their pros and their cons. And, given enough bourbon, we can probably be provoked to, go to the next slide and tell you what we really think about it. But these are areas, I think, of significant interest, and hopefully I've given you a sense of why there's such enthusiasm around this area in terms of the kind of biologies that can and are being driven. So just to summarize this, and then we'll move on to our panel discussion. Brief introduction to this concept of genome editing versus transgene delivery, whether it's ex vivo or in vivo. Genome editing gives one the ability to move in and look at knocking out an endogenous gene. It also gives one the ability to do targeted insertions within the genome and allows one to think about regulation of that targeted insertion with the native promoter or regulatory sequences. I think I've given you at least one perspective in terms of the issues relating to genome editing, targeted genome knockouts, and insertions in stem cells versus lentiviruses, the durability issues related to conventional AAV in vivo approaches, and then lastly, the field and how it's evolving. So that's an overview of this space. And why don't I ask our panelists to come up, and then we'll jump into a discussion around all of this.
My name is Bill Lundberg. I'm the Chief Scientific Officer of CRISPR Therapeutics. I'm trained as a clinician in internal medicine, hematology, oncology, and I've been in industry for a number of years in both small and large companies, running research and R&D, and most recently with CRISPR Therapeutics as the Chief Scientific Officer, building out research operations, which we have now established and up and running in Kendall Square, Massachusetts. Thanks.
Hi, my name is Jen King. I'm the Vice President of Business Development for Intellia Therapeutics, a CRISPR-Cas9 company that was formed in 2014. So we've been around for just over a year. Again, we're located in Cambridge, Massachusetts, right down the street from our fellows CRISPR. And we are based on foundational IP out of Jennifer Doudna's lab at Berkeley. And we're focused on developing potentially curative therapies for severe genetic-based diseases.
Great. So, Jen, let's start with you. I gave an overview of genome editing, conventional gene therapy. What parts of that would you like to subtract or add to?
Well, first I'd like to say I think there's space in the field for all forms of gene editing as well as gene therapy. I think they all have a place. I think gene therapy as well as the zinc fingers are a little more forward in translation into the clinic than the newcomer, CRISPR-Cas9, which was just published back in 2012. But I think there is some nice flexibility that the CRISPR-Cas9 platform allows for in terms of rapidly screening a number of potential sequences for the cut of interest, for the gene edit that you want to do. You can do this relatively rapidly because the targeting moiety is actually an RNA oligo, and the protein piece stays constant. And so you can rapidly screen a number of RNA oligos for their ability to cut specifically at the site of interest and generate the type of edit that you'd like to have.
Bill, anything you want to dissolve yourself or disengage from in terms of that talk?
Nothing to disengage from, something that I'm happy to reiterate. But first, let me say, I think you summed it up well.
Sorry, I didn't hear you.
I think you summed it up well.
Oh, thank you.
So it's also important for us to acknowledge, we're interested in making therapeutics, all three of us. We're in product-focused companies. And our ability to do that, I think, really rests on the gene therapy field that's come before us and the work that all of you have done in gene and cell therapies, particularly genetically modified organisms. And if I didn't have a really refined sense of how complex and how hard that landscape has been, particularly the regulatory landscape before the session this afternoon, I certainly have a finer appreciation for that now. So I think as we bring products forward, we really are able to leverage the great experience and expertise of many in this room and many in the gene therapy field.
So Bill, let's use that as a sort of a launching point to the I guess the main thing I'd like to talk about is in the area of genome editing, and obviously maybe from a CRISPR perspective, I'm happy to answer the same question from the zinc finger or talon perspective. What do you see as the major translational challenges for CRISPRs in the case of genome editing for therapeutics?
I think the major challenges are largely those in any really good development program. I think we need a disease to go after where we're confident that the target is really instrumental in pathophysiology, that there's good evidence that the modification of the target will lead to a therapeutic benefit or an effect on a biomarker surrogate or ultimately a clinical readout that we have confidence in, and that we can do good, solid drug development, preclinical development, manufacturing considerations, clinical stage development, regulatory strategy and execution to bring the product forward. Within the context specifically of gene editing, the elements that are unique, and I mean, both of you know this as well, is really in the context of making a double-stranded break in the DNA and having the cell repair that or repair it against an exogenous template to insert new DNA. The challenges there are really ensuring that we have robust efficacy and safety to the threshold that we need for a particular indication.
Great. Jen, you want to take on the same issue?
Yeah, I'd like to dive maybe a little more into the weeds because I think many scientists have brought CRISPR-Cas9 into their labs and have demonstrated that it's a robust technique. At least on the benchtop. And I think, it's incumbent upon the gene editing companies to really begin to industrialize that process. And I think there are going to be a lot of hurdles around assay development, reproducibility, manufacturing, et cetera, speaking to what you did. And I think the other piece is when I think about genome editing in general, I often think that there are a breadth of diseases we can address through genome editing, and it's really going to be limited by our ability to deliver to the cell type of interest, whether that's through a viral vector or potentially lipid nanoparticles or some other form of delivery. And then also in genome editing, you can go deep into a particular tissue by the type of edit you can do, whether you're knocking out a gene, whether you're inserting a gene in cis, or whether you're correcting a mutation. So when I think of gene editing, I think of both the breadth of all of the potential indications you can go after, as well as the depth by the different types of edits or changes you can make.
Great. It's a bit unfair given how long we've been doing this, but I'll give you at least our perspective on it. I'll try and say the following at the end, but in case I forget, we've been in the clinic with genome editing zinc finger nucleases for quite a while, and the most recent work, and the reason I'm saying this now is it's available to all of you, is a presentation we made to the NIH RAC, so the National Institute of Health Recombinant Advisory Committee, which is videotaped and audio as well, and it's online a couple of weeks ago. And I think the reason I mention that is it goes through in excruciating detail the kinds of issues that one needs to think about that are differentiated from a gene therapy approach versus an in vivo genome editing approach. And so there's detailed discussions of specificity. And remember what we're doing here, what our goal is, what we intend to do is break DNA, is break your DNA. Going in and making a double-stranded break. And so one of the critical regulatory safety questions is specificity. And so there's an enormous amount of emphasis from a regulatory and a development perspective on the specificity, the entire genome specificity of a genome editing therapeutic product. There's also obviously derivative of that, a lot of emphasis on toxicology and so on. So there's a granular discussion for those of you who are interested in what are the issues from at least a U.S. regulatory perspective, not necessarily an FDA perspective, but at least from an NIH RAC perspective on the challenges and differences of a genome editing approach. Maybe I'll answer the question first, and then you guys can go. I'm going to ask the question about sort of, okay, interesting platform, interesting approaches. What do you think are the areas of potential earliest translation, if you will? And I'll talk a little bit about where we are, but it'd be interested to know what you guys think. And if you don't want to name targets exactly, or you can, what do you think cell types wise, and that kind of thing, or directionally, what do you think people should be looking for in terms of therapeutic strategies or approaches? So for us, we've taken an approach both ex vivo in stem cells and T cells, as well as in vivo. The in vivo work I just referenced, and that'll be the newest work for us, looking at using AAV delivery of nucleases to the liver. But the work that we've done the longest now has been ex vivo. We've been in the clinic for 5 years in T cells, in CD4, CD8 cells, using genome editing strategies to knock out or disrupt the CCR5 receptor in an HIV setting. And then we're moving in with our partner Biogen into hematopoietic progenitors in the areas of beta thalassemia and sickle cell disease. So both ex vivo approaches and now more recently in vivo approaches. So you guys maybe talk a little bit about directionally what you think are the key areas of therapeutic development.
Yeah, I'd be happy to. I think it gets back to the earlier point around delivery. So Intellia is currently working in the ex vivo space. We have a partnership with Novartis that covers HSCs as well as CAR-Ts. And we're also looking at internal or in vivo delivery as well, initially to the liver, because there are methods for delivering nucleic acids to the liver.
I'm on the panel with two smart people. I don't have a lot more to add. I think we yeah, right. But I think we obviously pay close attention to what our colleagues at Sangamo have been doing for a number of years. Sangamo. Thank you. Sangamo have been doing for a couple of years, and the easiest and most proximate tissue to manipulate is bone marrow ex vivo, and there are a number of diseases for which one can address by accessing bone marrow and editing in bone marrow cells and reinfusing them. Two of those are HIV infection with CCR5 and targeting in one way or another hemoglobinopathies, which is what you're doing in your Biogen partner program. There are a series of other diseases in that space as well, primary immunodeficiencies, for which there are a number of therapies. Those diseases tend to be of smaller populations. And then beyond that, the way we think about indications in the context of diseases to think about applying gene editing to is really in the context of risk and risk assessment. And there's technology risk around the platform, and adding to that delivery risk is stacking a yet additional risk on top. So those areas for which gene therapy methods or other approaches have already provided evidence that delivery can work is a way to risk mitigate that. And that's the liver, as Jen was identifying, and also back of the eye, which another company is pursuing. So other areas where delivery appears to be currently tractable.
Great. So a few more minutes. We must have upset somebody in the audience. Any questions from anyone? Great. Can we get a microphone over here, please?
I'm from the CAT committee.
Yeah.
I would like to ask you two questions. One is scientific, another is regulatory. It took us many years to come down with some gene therapy products, and we recently approved Clybera. And then we might approve other things too. The scientific question I'm asking you is, when you say you compare the old convention technology using vector scaling genes with the gene editing, means to me that the technologies that you're using, they have some kind of leakage. They're not so specific always to specific locus. and you might have over there some problem specificity. Second question is related to the first, is the durability of an episomal vector delivering the gene, and how many generations down the line the cell will keep the gene permanently. And the last question, which is regulatory, how about risk assessment in all of this? That is major concern to us, at least to myself. Thank you.
You guys want to jump in? You want me to start? I don't care.
Go ahead.
I'm happy to.
Go ahead.
So thanks. Those are actually three really important questions, and not to sound like a regulator, but there's a lot of detail in here that depends on the specific context in which a program contains these issues. The issue of durability of the nuclease in the cell depends on how that nuclease is delivered. Many experiments, including animal models in which this technology is published, use mRNA to deliver the nuclease, or short RNAs to deliver, in our case with gene editing, the guide RNA components. And once either one of those components is gone, the nuclease has no more DNA cutting activity. In our hands and cell types.
But I think the outcome is permanent.
Right. The outcome of the edited cell is permanent for the life of that cell type. And in the case of bone marrow, if we were to edit bone marrow, either with zinc fingers or with CRISPR-Cas nucleases, those presumably repopulating stem cells would last, hopefully, the lifetime of the patient. But the specific nuclease itself would last a short period of time. In terms of risk assessment, you know, there is with this technology the possibility of off-target cutting, and that has to be weighed in a variety of different contexts. One is the risk-benefit to the patient in terms of overall disease. A second is, if you think about it in the context of the frequency with which double-stranded breaks occur in your genome with each cell cycle already. And there's a reason we all have non-homologous end joining mechanisms in the cell. That's because the DNA breaks. It has double-stranded breaks with each cycle of replication. We don't know how many because we can't measure it because it's below the limit of detection. But likewise, for certain zinc finger constructs and for certain CRISPR-Cas guides in particular systems where they've been tested, there are some that have no detectable off-target activity to the best of our ability to assess. But admittedly, our thresholds are here, and there's some uncertainty. Ideally, we'd like to be all the way down here. I think I covered some of those.
Yeah, no, you did. Jen, you want to add anything?
Just that the specificity for the different guides in the case of CRISPR-Cas9 are defined by an RNA, homologous RNA stretch, and can be really fine-tuned to ideally hit a single site in the genome. And the same is true with the zinc fingers as well. You can design the protein such that it hits only a single site in the genome, at least to the best level of our detection.
I'll be very brief and say I think the most important element is specificity, the ability to create reagents that are uniquely and singularly specific to their intended target. And the discussion here is added to that, but that I think is the critical piece. One more question here. I think one more question, then one more question, then we'll have to stop the panel.
How challenging is, or how stable are the constructs you're delivering in vivo, and how challenging is that? Or are you facing the same problems, double-stranded DNA, DNA decoys phased, or siRNA?
Yeah, so at least for the CRISPR-Cas9 constructs, really the protein, riboprotein, only has to do two things. It has to cut 2 strands of DNA. So stability is not something that you'd really want. You don't want something hanging around for a long time. So what we've seen is that they do clear, and it depends on the cell type you're using and the particular construct, but they do clear relatively rapidly. But at least in the case of gene editing, you know, that's almost desired, because the longer it hangs around, the more chance for it to potentially do off-target events.
But it also means they get trapped mostly or target the liver first, correct?
Correct, given our current delivery, yeah.
Okay. One more question here. Sorry.
Thank you. I have a question concerning the insertion of genetic material. Obviously, the concepts that are in the clinic now are all knockout concepts, and the efficiencies to which genes can be knocked out are quite impressive now, I think. The efficiencies of or repairing a gene by insertion of genetic material is, to my knowledge, much lower. Are there concepts, or is there progress on that field, and how can that be achieved?
I'll start, and Bill, maybe you should jump into this. I think your sense of proportionality is absolutely correct. I mean, if one can achieve 80% knockout in CD34 cells, one can receive 10% of targeted integration in that same cell. I mean, order of magnitude kind of thing. So it gives one, and Bill was talking about this earlier, you focus on the kinds of targets where 80% or 50% knockout can achieve your desired biological outcome. You don't look at targeted integration opportunities where you need 80% targeted integration to have a therapeutic effect. I mean, anybody want to comment further?
Were you going to ask? Were you going to clarify something there? You look like you had a comment.
No, I just thought, okay, well, in the knockout field, you started out with efficiencies of 5, 10, yep. 20%, and now you're at 80. In the insertion field, people started out at 0.1%, 1%. You say now you're at 10. Is 10, you think, the limit, or will you be at 80 sometime as well?
Yeah, I'm happy to speculate on that. I think it's easy. In the absence of data, it all looks good. There have been a number of publications around different approaches and different methodologies for modifying the endogenous DNA repair mechanisms, DNA ligase 5, for example, or other proteins that participate in non-homologous end joining. And one can bias that either by modifying the repair systems in the presence of a homologous repair template, or by just doing a broad small molecule screen to identify small molecules that impact that. And the numbers have been going up. I haven't seen anything that suggests that we're at a plateau. But again, it also depends very much on the cut, the piece of DNA being inserted, the cell type being manipulated, and bringing all that into the animal or into the person. I think we have to see.
Great. I'm going to, one last question in the back, and I promise we'll stop. If it wasn't for this being my favorite subject, we'd have cut it off a long time ago.
Thank you for the chance. My question is related to how you do the development. All of you mentioned the possibility of doing in vivo delivery. So being these genome-specific reagents, how do you plan your toxicological studies? I imagine in some cases you can make sequences that are shared by, for example, mouse or rat and human genomes, but in many other cases you might not.
Yeah, it's very, very important. And so we build species-specific reagents for a given target, for toxicology studies, even including non-human primate studies where they're not homologous with the target sequence that we're going after. So it does make development more complex.
It's a complicated topic. I mean, I think when you think about an ortholog that is now a mouse reagent, you have to ask yourself what really is the relevance for on or off target data supporting a regulatory filing. I guess you could have some evidence of on-target unintended effects from your gene edit. Will the edited cells in the liver still work like liver cells? But the off-target effects are much more complicated. There is precedent in the development of antibodies that do not cross-react with any lower species, and regulatory pathways that have been undertaken for those types of molecules. And that's an analogy of what we're trying to do here.
Great. Thank you all very much for your questions. Thank you to the panelists. Very enjoyable. Thanks.